Welcome back. The ease of access to this cutting-edge technology is part of the democratization of biology. Websites like Jove, the Journal of Visualized Experiments, provide thousands of how-to videos on the experiments of bioscience. There are weekend hobbyists, self-styled as biohackers, do-it-yourself biologists from around the world who have been carrying out the sorts of experiments that until recently were only possible in professional labs. This movement has become possible because the all-important PCR machines for amplifying large amounts of DNA from tiny little samples can now be purchased cheaply online, while enzymes and chemicals for creating, manipulating and sticking together DNA can be ordered off the shelf. Here's a centrifuge designed by Carlin Ye for biohackers. If you go to tnd.com, you can get the kit for this for just $260. You can also buy a lot of stuff from the medical lab equipment subcategory of eBay's business, office and industrial section. Or with the lab-equipped with cheap equipment, you can clone and sequence a gene in a couple of days using freely downloadable protocols. Biohacking has gone mainstream with biohacking organizations, biohacking events, virtual networks and co-working spaces for enthusiasts to swap stories, advice and protocols. Visit one of the many online forums dedicated to the field, such as BioCurious. You'll find thousands wanting to join the movement, all eager to try and engineer DNA in their kitchen or garage labs. So high school students and others can learn about genetic engineering by introducing a gene that makes the fluorescent protein for this jellyfish into bacteria to make them glow in the dark. In 2008, 3 scientists won Nobel Prizes for this work. Already, it's literally child's play. But many biohackers don't just want to play with glowing bacteria. Instead, they share an impulse to empower themselves and to not leave everything to the experts. An example of this is Kay All. All built her own genetic testing kit in a small room in Cambridge, Massachusetts. In her makeshift lab, she analysed a specific gene mutation linked to a disease her father was diagnosed with, called hemochromatosis. If you have it, the body is unable to get rid of excess iron. Well, with relatively simple, cheap tests, all tested her own DNA and found both the faulty gene from her dad and the unaffected one inherited from her mother. The result meant that she's a carrier, but unlikely to contract the disease herself. Well, biohacking is no longer just confined to people's houses and vintage equipment. There are so-called biohacker spaces, such as GeneSpace in Brooklyn, New York. GeneSpace is a state-of-the-art DIY biolaboratory where everyone can experiment for a fee of $100 per month. It has a motto: remember the time when science was fun. Well, there's a bunch of students at GeneSpace. They look like they're having fun, don't they? The ideas, the energy, and the drive behind these people are reminiscent of the early computer pioneers who built the software and hardware that kick-started the computer revolution and went on to found household names like Microsoft and Apple. So it's not too great a stretch of the imagination to see that the next generation of entrepreneurs can be biologists, not programmers. In fact, Microsoft's founder, Bill Gates, told Wired magazine that if he were young today, he would be hacking biology, creating artificial life with DNA synthesis. Creating artificial life with DNA synthesis is similar to a machine language program. So Bill Gates added, if you want to change the world in some big way, that's where you should start. Biological molecules. Well, Bill Gates' comments got a lot of publicity, and as the field has grown, so have the headlines. Some have been positive, trumpeting its promise. Others have been much less welcoming, fearing a possible dawn of bioterrorism. Linked to this lecture is a TED Talk called Biohacking, You Can Do It Too, by Ellen Jorgensen, who opened GeneSpace. Jorgensen talks enthusiastically about her DIY lab devoted to citizen science, but she also makes a point of her contacts with the FBI and that she doesn't work with pathogens. She says that if you work with pathogens, you're not a biohacker, you're a bioterrorist. While a specific case of weaponizing engineered life forms is often put forward in opposition to both genetic biology and synthetic biology. A synthetic biology expert, Rob Carlson, says it's easier to fixate on the threat than embrace the opportunity. But it is important to acknowledge that there is a threat. The November 2012 edition of The Atlantic featured a scenario to assassinate the United States president in 2016 by using a bioweapon specifically tailored to his genetic makeup, a virus that targeted the commander-in-chief and no one else. The authors, including genomic futurists Andrew Hessel and cybercrime expert Mark Goodman, acknowledged that the plausibility of a hit on the president by 2016 is far-fetched. But they did point out some worrisome dual uses of the free accessibility of knowledge training and commercial DNA synthesis. Thanks to advances in genetics, we now know that each cancer is unique, and research is shifting to the development of personalized medicines, designer therapies that can exterminate specific cancerous cells in a specific way, in a specific person. Combine those technologies with those used by Ventnor, and it could, in future, be possible to engineer a pathogen targeted against a single person. And the Atlantic article makes the point that the attack organism wouldn't necessarily be designed to kill. It may, for example, be designed to change a person's serotonin or cortisol production to wreak havoc with their ability to negotiate. The potential for personalized bioweapons is some way off in the future, and will probably still require considerable resources and be beyond the technical acumen of even the best amateur bioengineers. That means sponsorship by a corporation or government. However, engineering bioweapons of mass destruction might not require as much finesse. Rogue biohackers could potentially manipulate toxic genes to create dangerous pathogenic microbes. Is it then justifiable to fear that amateur biohackers might see it as a personal challenge to make a killer virus in their garage? That's how Michael Osterholm of the United States National Science Advisory Board for Biosecurity put it in 2012. Is every second-hand biotech machine sold on eBay a step in that direction? Well, the FBI clearly sees it as a possibility, and it's perhaps easy to see why. Take the recent controversy surrounding man-made forms of the flu virus. Again, acting on Feynman's maxim, what I cannot create, I do not understand, two teams of scientists mutated the H5N1 bird flu virus in such a way that it could spread between mammals more easily. Well, they were trying to understand how natural viruses evolve into more dangerous forms so we can arm ourselves against the certainty of it happening naturally. But their announcements sparked an international storm. Should the work be published in full to help authorities prepare for a potential pandemic, or should it be withheld from the research community to prevent possible misuse by amateur scientists or even bioterrorists? As a result of months of pressure from scientific journals and notable names in the field, the work was eventually published. On the grounds that the most productive and creative science comes when information is shared freely, and that in doing so we equip ourselves to tackle the threat should it come at all. Well, the knowledge and technology used to mutate the H5N1 bird flu virus was way beyond what amateur DIY biohackers do today. Nevertheless, the FBI's Biological Countermeasures Unit is keeping close tabs on biohackers. Agents in 56 FBI offices across the United States have reached out to the field's leading names to learn about what they do. And the unit's leader, Nathaniel Head, organised the first international DIY bio outreach workshop in June 2012. Head said, "We want to learn about biohacking and to be able to separate between the white and the black hats." Well, that leaves no doubt as to the reason behind the workshop. The FBI want means of differentiating between harmless amateur tinkerers and potential bioterrorists misusing genetic engineering technology. Perhaps the ultimate application of recombinant DNA technology is gene therapy, the direct transfer of genes into humans. The concept of gene therapy is relatively straightforward. You identify diseases that result from mutated genes, and you replace the defective genes with working copies to correct the ailment. In practice, it's a little trickier. If we try to introduce naked DNA on its own, it's very inefficient. You need to inject very high levels of DNA, and the DNA doesn't last very long. Gene therapists usually rely instead on the natural ability of viruses to insert their DNA into their host genome. Viruses are nature's way of introducing DNA into cells. You use a virus that doesn't cause a human disease. Every virus has an envelope made of protein that determines when the virus likes to go in the body. And inside the envelope is the virus's own DNA that encodes the genes that make baby viruses. For gene therapy, you take the virus genes out and insert the treatment genes, usually good working human genes. Then you use the envelope to deliver the genes where you want them to go. So, for example, into the heart muscle, if you're using an engineered virus to heal damaged and struggling hearts. So it's like a reverse Trojan horse. It's a nasty thing on the outside, but it's actually delivering something good on the inside. Well, there's a new technique that looks set to replace viruses called CRISPR. CRISPR uses a customizable RNA to guide a DNA cutting enzyme to the right spot in the DNA. Geneticists have been astounded at how easy, accurate and effective CRISPR is at altering the genetic code of any life form. And they believe it will soon revolutionise the study and treatment of a range of diseases, from cancer, incurable viruses such as HIV, to inherited genetic disorders such as sickle cell anemia, Huntington's disease and Down syndrome. There are already over 1,900 gene therapy clinical trials, many of which look promising, and CRISPR is just the latest technology and the easiest to do. But CRISPR and some related techniques are enabling, suddenly, hundreds of researchers to advance things. And that kind of open sharing is a very efficient method of getting results. The gene therapy conducted to date has targeted only non-reproductive cells. This is called somatic gene therapy. It's only legal when used to correct serious illness. Correcting a genetic deficit in body cells will not affect the genes of future generations. In germline gene therapy, sperm or eggs are modified by the introduction of functional genes, which are integrated into their genomes. This would allow the therapy, to be passed on to later generations. But fears over its safety and the prospect of so-called designer babies led to germline gene therapy being made illegal in many countries. Well, the new CRISPR gene editing technique could address many of the safety concerns because it's so accurate. Some scientists now believe it's only a matter of time before IVF doctors suggest it's used to eliminate genetic diseases from affected family by changing the DNA in an egg, sperm or embryo. I'll leave you with this quote from Dagan Wells.